At the peak of harvest season in places like Uganda's Kayunga market, pineapples and mangoes are abundant and cheap. But within a matter of just a few weeks, those exact same fruits soften, rupture, and collapse into mold. This rapid decay occurs because fruits are essentially biological water balloons packed with natural sugars. They are biologically programmed to decompose, providing the perfect nutrient-dense environment for life to break them down. The economic consequences of this biological flaw are severe. As surplus harvests rot in the rubbish bins, the supply drops to zero, and the price of any remaining off-season fruit skyrockets. Rotting is a specific physical vulnerability. By understanding the mechanics of how fruit breaks down, we can systematically engineer against it to preserve both the food and its value. Decay originates from two distinct fronts. The internal trigger comes from the fruit's own intrinsic enzymes. These proteins actively break down cell walls to soften the flesh, accelerating over-ripening and eventual structural collapse. The external threat comes from ambient microorganisms, specifically bacteria and yeast. They detect the water and sugar, breach the weakened cell walls, and rapidly multiply. Allowing these microbes to multiply unchecked directly leads to food poisoning. Consuming contaminated fermenting fruit introduces these pathogens and their toxins into the human digestive system. The entire science of fruit preservation is dedicated to one goal: engineering an environment entirely hostile to microbial life. Every processing technique we use is a deliberate scientific countermeasure designed to disable enzymes, destroy bacteria, and lock the fruit into a safe, shelf-stable state. To build a hostile environment, we have to eliminate the primary requirement for microbial survival: free liquid water. This cross-section of a fruit inside a solar dryer illustrates the most basic physical countermeasure. By using solar heat to force water molecules to evaporate into the air, we systematically remove the moisture that bacteria require to maintain cellular function. But many preserved products, like squashes and jams, require a wet environment. To safely maintain high moisture, we apply extreme heat. In this diagram, zooming into the boiling fruit pulp, intense heat actively unfolds and denatures intrinsic enzymes and invading bacterial proteins, halting biological activity immediately. Boiling and dehydration neutralize immediate threats, but leave the food physically vulnerable to future contamination the moment it cools down or is exposed to air. The high concentration of sugar used in jam production provides a mechanical defense. It functions as a lethal biological barrier. This relies on osmosis, the biological principle where water naturally moves across a cell membrane from high concentration to low concentration. In this diagram, an intact bacterial cell is suspended in an environment densely packed with sucrose. Because the exterior sugar concentration is vastly higher, it forces water out of the pathogen. The cell membrane visibly shrivels and collapses entirely. High sugar content acts as an invisible osmotic barrier. It systematically dehydrates any invading pathogens before they can multiply and spoil the product. While sugar provides defense, it doesn't provide structure. Simply boiling fruit and sugar together does not yield a thick jam. It yields a watery syrup. To lock that syrup into a stable gel, the mixture requires a structural polymer called pectin, a natural starch found in fruit skin and seeds. When processing low-pectin pineapple, added pectin chains float freely, repelling each other due to negative charges. Adding citric acid neutralizes these charges, allowing the chains to snap together into an interlocking 3D matrix, trapping the syrup into a rigid gel. To visually verify this exact thermodynamic phase change in the field, preservers perform the wrinkle test. On a cold plate, the rapid temperature drop forces the pectin to set. If the surface wrinkles under a finger, the chemical bond is achieved. A successful jam is a carefully constructed chemical matrix. The pectin provides the architecture, while the sugar protects the interior. Concentrated liquid fruit squashes present a different challenge. Because they must remain pourable, they lack the dense, high-sugar osmotic structure that protects jam. Approved chemical preservatives provide a critical safety mechanism against highly dangerous organic threats. These structural chemical formulas show sodium benzoate and potassium sorbate. They are targeted molecular inhibitors used specifically in liquid preservation. When added to fruit squash, these chemicals neutralize yeasts and molds that survive the liquid environment, preventing severe fermentation and dangerous food poisoning. Because these inhibitors are potent, they must be measured strictly to approved health standards. The correct dosage ensures the product remains completely safe for human consumption while totally inhospitable to mold. When physical and structural barriers are absent, exact chemical intervention is the only scientific method available to prevent lethal contamination. By applying these principles, a single raw fruit harvest can be engineered into entirely different shelf-stable products, depending on the chemical pathway chosen. This flowchart tracks the pipelines. Boiling crushed pulp with pectin and sugar forms jam. Extracting clear liquid juice with gelation chemistry creates jelly. Expanding downward, adding bitter citrus peels yields marmalade, while diluting juice with chemical stabilizers creates squash. Regardless of the final product, the engineered defenses are only permanent if the food is sealed in completely sterilized glass jars, maintaining a strict hygienic barrier against the outside world. Varying the physical components of the fruit, whether it's the pulp, the peel, or the clear juice, requires distinct chemical interventions to safely reach preservation. We've examined 4 active defense vectors: thermal destruction to kill immediate threats, osmotic stress to dehydrate surviving cells, structural gelation to bind the moisture, and chemical inhibition to prevent fermentation. Mastering this specific chemistry is a requirement for meeting rigorous public health standards. It is the only reliable way to ensure that stored food does not become a vector for disease. By applying these biochemical principles, local farmers can capture the immense value of a fleeting harvest. They transform highly perishable, rainy season fruit into safe, shelf-stable commodities that generate reliable profit all year round.